Hi, sir.
Hello.
Yeah. Thank you, sir, for joining.
Relevance. The clinical relevance is like, that if you consider the application of biomaterials from head to toe in any human patients, for example, if you go from the head, like, cranial prosthesis, everybody's head at certain shape and cranial has some shape and size. So therefore, any reconstruction of cranial defects, you need to have more patient-specific implants. What I mean that one single size does not fit every patient. So therefore, and then it is not only size, but also shape and morphology of the defects is very different from patient to patients. So therefore, we need to have patient-specific implants as far as the cranial implants is concerned. Now, if you go to dental, again, whenever you are considering the dental implants like titanium screws or zirconia screws, depending on the bone conditions and depending on the jawbone cavity that where implants is to be made, you need to have a different sizes of the dental implants. When you go to acetabular line and acetabular implants, there again a range of sizes are important. So if you look at this additive manufacturing, its current market value is $1.2 billion US at a compound annual growth rate like CAGR of 29% in India. So if you look at that, what are the key attributes of the 3D printing? So one is the patient-specific implants. It also gives us a lot of freedom of design, like you can design the implants the way you want. And as a result, because it will essentially fit into the anatomical space or anatomical locations of the human patients, it will certainly give you improved functionality and enhanced biocompatibility. In the context of India, these are the growth drivers like aging population in India, expanding healthcare access and quality, and health tourism by many patients from Africa, by many patients from several other developing nations in Indian subcontinent who come to India for better healthcare, but an affordable price. So not only better healthcare, but also affordable price. In addition to 1.6 billion people in India, we also have significant number of medical tourists. That is the point I was trying to make. Now, what are the restraints here? In the restraints is certainly unaffordable healthcare costs because of the most of the implants are imported from North America and Europe, and also intense competition in the orthopedic market. So this is just some materials-based example of the additive manufacturing of bone implants. If you look at the top row, it is a calcium deficient hydroxyapatite, CDHA, which has been placed in the particular side of, at the particular bone defect. And then bottom, if you see the struvite cement, in the struvite cement, it degrades with time, like 4 months, 7 months, and 10 months. And if you look at this CDHA, calcium deficient hydroxyapatite, it stays there because it's not biodegradable. So compared to calcium deficient hydroxyapatite, it is a magnesium phosphate-based materials, which also allows you to combine good stability with high strength and good bone regeneration. Now, just 1 second. Just 1 second. Yeah, so in terms of the science of the powder binder interactions, what you are going to see here that we have been using this 3D binder jet printing for quite some time now. And in this 3D binder jet printing, what we are trying to understand that not only that we'll develop our understanding of the capability of binder jet 3D printing to develop different materials, not only ceramics, but also metals, but also how the binder interacts with the powder bed. And in terms of our understanding of the binder interaction, the powder beds of what you see in the top left panel, It is a drop-on-demand type of 3D printing, what we call DOD, and it is inkjet printing. Now that every time that from the feed bed to the build bed, one layer of powder is displaced, the binder jet also impacts on the powder bed, and then it penetrates into the powder bed. We're trying to understand that, what is the time scale of these binder material interactions, and also what is the spatio-temporal kinetics. Spatio-temporal kinetics means how the binder spreads into the powder bed, what is the time scale it will take. And in terms of the fluid properties of the binder, which is important for its printability, what you see is the two dimensionless number from the classical fluid dynamics. One is the Ohnesorge number and one is the Reynolds number. Now, for every binder, we need to find out that what is these two dimensionless number. And the combination of the dimensionless number should fall within the hatched region, as I have been showing here in the top right-hand plot. It is and if this combination of these two dimensional number falls there, then that particular binder, you can use it for printing these materials. And therefore, the other parameters which are of importance, not only the binder characteristics, but also powder characteristics like powder size distribution, powder shape, flowability, that is very important for some of the ceramic powders that I'll show you later, particularly in the context of zirconia. Then post-processing, and then process parameters like what is the layer thickness, what is the binder saturation, and so on. And the right-hand down in the right-hand left panel at the bottom, you can see that when the binder drop impacts on the powder bed, how it comes, how it dissolves, and then how it spreads over time. So few years back, one of my students who is currently postdoc at Cato Lawrence's lab in US, Remanto, has done some thoughtful experiments where he has mixed the titanium 6 aluminum 4 vanadium powders with certain initiator. And in case of the binder, what he has done, he has put this monomer and accelerator together and make a monomer solution. Now, when this monomer solution impacts on the binder on the powder bed, what will happen, what has been shown, what is the bottom panel, that as soon as the binder interacts with the powder bed, there's a in situ polymerization form triggers. And if due to the in situ polymerization, there's a chains that will form, and these chains will try to kind of hold these powder particles together, so it will have much more robust binding action. But as I said in the last few minutes, that one has to find out that what is the Ohnesorge number, what is the Reynolds number. We have done experiments while these particular binder flows through the orifice. Then we put some high-speed camera and trying to find out that what is the binder droplet distribution, size distribution, what you see here in this particular movie. And you can see that, that this is how the binder particles, they come, they impact. And then through this high-speed camera, you find out that what is the binder droplet distributions. And from there, you can find out that what is the Ohnesorge number and Reynolds number. Because many other things you can find out simply from the chemical, physical properties of the binder. Now, titanium 6 aluminum 4 vanadium as a model material, because it has been very widely used for biomedical applications, we have been able to show that binder jet 3D printing can produce at least like 72% dense materials with 98% interconnected porosity. And this material, because it is porous, it also has very compromised mechanical properties. And here we have found out viable modulus is around 8 and strength is kind of close to 100 megapascal. And since we have used specific binder, we try to find out that whether it has any negative impact on the cell compatibility, cytocompatibility. We have used fibroblast and osteoblast cells, and then we have found out that binder jetted titanium 6 aluminum does not have any negative impact on the as far as the cell proliferation is concerned. So this work was published in 2019 in the journal Biomaterials. Now, coming to the science of binder powder interactions, we have gone into much more details. We have done synchrotron experiments, and because a few research groups in the world particularly Denisuk as well as Popovich groups, they have done some experiments where they have used the droplets of around 2 millimeter to 6 millimeter diameter. But however, drop sizes in inkjet printing varies much higher, much lower, that is 10 to 80 micron. And dynamic monitoring is crucial because that is, you can find out the real-time ink powder interactions. We have done some collaborations with Brian Darby's group at University of Manchester, where we have used Diamond Light Station in UK. And then these experiments were conducted in UK, but all the planning as well as the experimental plan was done by our group. So what we have done, we have 3D printed several small cassettes-like structures where we have put alumina powder, and from the top piezoelectric actuator, actually it supplies through 60 micron orifice the ink droplets in a manner which simulates very closely that of drop-on-demand printer. And in this particular case, that ink was made of ethylene glycol and deionized water, and this has viscosity properties which is fairly close to that of our binder that which we have used. So what you see on the right-hand side, that in the detector, the 2D projected X-ray radiographs were acquired and they were subsequently analyzed. Now, in all these synchrotron experiments, they essentially produce huge amount of data, and this data is of the size of terabyte data. And this data analysis is one of the key things as far as our understanding goes. And this data was very, very carefully analyzed and using several softwares. And then finally, we got an understanding on the kinetics of the infiltration dynamics. And if you see that some of this is the typical 2D X-ray radiographs you get, and then you can see that what is the weighting contour, what is the penetration depth, and what is the surface speed. And from there, you can find out that what is the interaction volume or penetration volume into the powder bed. And this is the impacted droplet and is spreading both in surface and volume. And there are some established equations and model. For example, Denisuk's model of the total penetrated liquid volume can be found out by this specific equation. Now, when we plot this weighting volume experimentally measured as a function of time and time scale, you can consider it is a less than 1 second. So in this particular very small time scale, when you have plotted this weighting volume versus time, it follows a curve which is fairly close to that computationally predicted plots. And there is some upper bound and lower bound, like many of these physical processes, we always find that there is always upper bound or lower bound, and somehow our experimental results is somewhere midway between the upper bound and lower bound of predictions. This is the summary of what we have understood during this synchrotron study, and you can see that how progressively weighting contour spreads in both Z directions and also in the lateral directions. And therefore, interface is continuously moving more along the Z direction. And a time increment here, you can see it's 0.1 second. So this kind of very fine scale experiments were certainly very useful to develop our understanding on the science aspect of the ink powder interactions. And this was published in our Essays Applied Materials and Interfaces. Now let us move on to discuss some of the materials aspects, what kind of materials we can produce in the 3D binder jet. Now, strontium sulfate magnesium phosphate certainly was of interest, and this was part of our one collaborations a few years ago with University of Würzburg in Professor Wei Gebruek's groups. And there, primarily, the idea was to use some magnesium phosphate, particularly strontium sulfate magnesium phosphate. The question is, why magnesium phosphate? Why not calcium phosphate? The answer is that in case of calcium phosphate, in case of magnesium phosphate, it has higher biodegradability with respect to calcium phosphates, and there is also absence of low soluble phases like hydroxyapatite. Now, in terms of properties, if you look at this, there is a company called Inoter that is based in Dresden in Germany, and then you can see that Inoter has produced 3D printed calcium phosphate scaffolds where the property is somewhere around 5 to 30 megapascal. For the magnesium phosphate cement, which is used as aqueous paste, where the properties were much higher, like 81 MPa and so on. Now, in the specific case of strontium sodium magnesium phosphate, we started with using 3 different precursors. One is a magnesium hydrogen phosphate, magnesium hydroxide, and strontium carbonate. And there is a formulation that we have used to 4:1:1 or 2:1, depending on what is the targeted final composition. We use the binder as a deionized water, and then after the printing is over, we have used 2 post-processing, or either sintering or chemical conversion. In chemical conversion, we have used 3.5 molar diammonium hydrogen phosphate solution for 1 hour. So in terms of the phase assemblage in the 3D printed ceramics, we have found out that there are 2 phases that is coming up very fast. One is a ferritinite, that is the Mg3PO4 and one is a struvite. So depending on what is the amount of strontium substitution, now the relative amount of these phases also vary. In all our 3D printed structures, we try to emphasize that how the 3D microstructure develops, and what is the interconnectivity or interconnected pore structure. And in the left side, you can see magnesium phosphate that is sintered, and the right side, you can see that converted, that chemically converted. And what you see, this typical pore size distribution, it follows more like a log normal type of distribution, and percentage porosity is somewhere around 15 to 22%, 15 to 20%. Now, since it's a porous microstructures, one of the major properties that we emphasize is the strength properties. And the strength properties, it is not only the absolute value of the strength, but also strength reliability. And strength reliability, typically we measure using the viable statistics or weakest link fracture statistics, and there you get a value which is called viable modulus. And this viable modulus value in our particular case varies around 5 to 8 or 9. Higher the viable modulus, better is the strength reliability, because there will be less spread of the strength values. So lower the viable modulus, more unreliable is the strength properties. Now, if you look at this particular table, most of the traditional ceramics or even alumina, silicon nitride, their viable modulus varies around 5 to 10, and they are used in very engineering applications. And sometimes, alumina is also used for biomedical applications. So what I'm trying to emphasize here is that these magnesium phosphate-based materials or strontium magnesium phosphate-based materials that we have developed in this particular case, they have viable modulus which is comparable to many of the engineering ceramics. Since strontium is present, our natural intention was to find out that, that how much strontium dissolves or in the solution. And therefore, we have done these experiments, in vitro dissolution experiments, where we have found out that with increasing amount of strontium doping, strontium ion is is leached into the solution. And particularly with Mg2SR1PO4 that strontium percentage is certainly higher than the other composition. So this is the summary of what I have been telling for last 10 minutes or so, that we have found out that these materials is reasonably modest strength reliability, and also these materials have very passive degradation behavior in DMEM. DMEM is the cell culture medium. This was published in Acta Biomaterials. Right. So apart from this, we have also done some other studies, that is on the zirconia. In case of zirconia, it was quite challenging because we tried to use different zirconia powders, and we are trying to find out that whether similar binder can be used in this case of binder jet 3D printing of zirconia. So to start with, we mixed this zirconia 3 more % ultra-stabilized zirconia with zirconium silicate, that is ZrSiO4 8H2O, and we mixed with initiator, that is APS initiator, and zirconia to zircon was mixed in 3:1 ratio. Then after that it was sieved, and then we have used the binder jet 3D printing. And when we have not done any other modifications in the design stage, we found out that there is a core shell mode of printing. Core shell mode of printing means in the central region it is much more dense, whereas outside the central region, it is more porous. And this is one of the major drawbacks for this kind of 3D printing of bioceramics. We are trying to find out that how, and then one of the major reason is that powders. So now question that should come to your mind that why zirconia cannot be printed alone and why do you need to mix with zirconium silicate. Now, binder powder bed packing is one of the major thing in this binder jet 3D printing. Now, if your powders are extremely flowable and very fine distribution, these powders are not good for particularly binder jet 3D printing, which we learned with experiences. However, there we have found out that binder alone cannot bind or trap particles, which are not packed cohesively. So in order to ensure cohesive packing of the powders, what we have found out, we mix with zirconium silicate so that it is not very much flowable, but also at the same time, it allows good packing here. Now, here is the trick. What we have adapted that during the CAD file, we want to bypass the core shell mode of printing. So any three-dimensional structures, you can imagine that it is composed of capital N number of layers. And these N number of layers, depending on what is the cross-section with the cylindrical cross-sections or rectangular cross-sections, you can have this kind of N number of parallel layers. And in each N number of parallel layers, you can introduce some slits in each of these 2D layers. And what you see in the next slide, that earlier I mentioned about the core shell printing, and you can see in the top left figure, there is very clear presence of the core and shell. But once you start the discretizing or segmenting each of this, each of the 2 layers, then what we can ensure highest saturation level during the binder jet 3D printing. For example, if you look at this schematic, the CD, that is the intra-segment bleeding, like when the binder jet is impacting, whereas E and A, that is the inter-segment bleeding. So each of this segment, if you go to the higher level, you can ensure that binder bleeding and joining actually takes place. That leads to more homogeneous infiltration, more homogeneous printing. This has been monitored in the real-time sense to ensure that there is no core and shell effect in the case of this zirconia printing. So one of the things that we have learned is that you can do infiltration of this unstabilized zirconia as well as the stabilized zirconia, 3 mole% intra-stabilized zirconia, and then this infiltration can be done with the precursor salts. And these precursor salts either can be zirconium oxychloride or can be yttrium nitrate. And then what we expect that during the infiltration and post-infiltration sintering, zirconium oxychloride infiltration will be converted to tetragonal zirconia, and this tetragonal zirconia during sintering, if you don't sinter them at appropriate level, then it can be transformed to monoclinic zirconia, which one should avoid. Now, in the next step, what we have done, not only zirconium oxychloride, but also we have used yttrium nitrate solution. And once we have done this infiltration, we have found out that after sintering, it will form the yttrium-stabilized zirconia, particularly 49 degrees of 5 years, and monoclinic phase into crack formation that we have seen in before, that can be carefully resolved. Now, in terms of the microstructure, what you have found out that core shell mode of microstructure on the right-hand side panel, the particle panel, you don't see any stark difference from the center to the edge of this structure. But what you certainly notice, there is still microporous structure. And this microporosity is something which is inherent with the binder jet 3D printing, which you cannot avoid in real life. But certainly in the left-hand side image, you can see there's unsegmented core, and this unsegmented core has much inhomogeneous porosity. This reduction in the large porosity or agglomerate pore agglomerates, that is also reflected in this getting little bit higher viable modulus. And this viable modulus, if you look at in the core shell structure, is 3.2. In the without any core shell structure, it is 3.8 as far as the flexural strength reliability is concerned. Same thing we have found out in case of this slightly higher viable modulus when we measure the compression strength. This is just recently published in Journal of European Ceramic Society. Now let us move on to the 3D extrusion printing. As I said in the beginning, it is biomaterial printing, not the bioprinting. So there is a distinction between these two. I'll slowly explain. Now, Brian Darby, one of our collaborators from University of Manchester, wrote this paper in 2012 in Science Journal, and where he has told the scientific community that it is possible to print natural tissue-like structure in an artificial manner by bioprinting technique. And this one of the things that he has shown, he has published in that paper is that, for example, in case of the cartilage model, you can use that one specific biomaterial ink where chondrocyte cells can be thrown in, and these chondrocyte cells is cartilage cells. Then after curing, as you can see that HNU is that is essentially curing, you get a cartilage-like structure where you can have an implant. Around the implant, you have cartilage formation in vivo. Now, what are the different ways that you can do this bioprinting or biomaterial printing? What you will see in next 15 or 20 minutes is that extrusion-based bioprinting, where you can either put pneumatic pressure or piston or screw-like structure, so that it will force the biomaterial to go through the orifice. There is another way of doing things is that inkjet-based bioprinting, and third one is a laser-based bioprinting. The laser-based bioprinting also you can do in this stereolithography. Now, one of the major properties of this 3D extrusion printing is that polymers. What polymers that you use, what are the fillers you used, and whether we use any cells to it, or whether you are trying to position the cells within the biomaterials so that when you are printing these materials, the cells are also placed in these artificial tissues. The shape fidelity is one of the main things. Shape fidelity, there are a couple of important terminology that I'm going to slowly explain you. One is called printability, one is called buildability. So printability essentially means given a specific hydrogel composition or given a specific polymer scaffold composition, how that particular ink can be printed without nozzle clogging, and that can be printed into complex porous architecture, and also that it can be printed in a relatively larger volume structures. And buildability essentially means that how you can build a longer structures where the shape fidelity is maintained and whether the shape can be retained without, or in other words, that longer structure, complex structure can be built using the same ink without any shape distortion. So a buildable ink, an ink which is good buildability must have good printability. But the reverse is not true. What it means that a printable ink may not have good buildability. I hope I have been able to explain you that what is the distinction between the printability and buildability. So what are the three important aspects that you need to develop an understanding, particularly in 3D extrusion printing? What is the printing related parameters, like, certainly what is the pressure and what is the nozzle size that you need to do. And then is the gelation. Now, gelation, you can see here that three different things has been mentioned in gelation. One is the UV cross-linking, one is the thermal cross-linking, and third one is the chemical cross-linking. So many times people use that calcium chloride solution that we expect that calcium +2 ions helps in the chemical cross-linking. People use barium chloride solution also with the expectation the barium +2 ions help in the cross-linking. And there's the UV cross-linking that is very popular. In terms of the rheological properties of this biomaterial ink, what is most desirable is the shear thinning behavior. What you can see, viscosity is as a function of shear rate, that as we increase the shear rate, viscosity should go down and slowly going off slowly show the non-Newtonian kind of behavior. And there should be a sharp gel transition. So you can see on the right-hand side that at the gel point, but this transition should be extremely sharp, not like a very delayed kind of transition. As I said a few minutes ago, that there are two parameters or two aspects which are important. One is the buildability, one is printability. And you can see that in order to enhance the buildability, you have to increase the layer height, flow, and build rate. But in case of the printability, the biometal ink should have good gel strength, and also temperature of printhead has to be very carefully monitored, and printing time also should increase. So these are the two things that is one has to have a good compromise for biomaterial ink or bio ink to be most successful in the particular clinical translation. Since next 15-20 minutes, I'm going to show you our work related to the gelatin methacrylate based biomaterial ink. I would also like to tell you at this point that why gelatin methacrylate based biomaterial ink is important, because there are significant work has been done, and then scientists have speculated that gelatin methacrylate based biomaterial ink can be carefully tailored. Their composition can be tailored to construct different artificial tissues, not only for skin, but also for nerve, neural regeneration, bone tissue, liver, etc. Just to give an example of the osteochondral tissue regeneration, this is the particular case for the gelatin methacrylate with secondary additive PAM. And then when people have used this particular biomaterial ink, they have also grown chondrocyte cells. And these chondrocyte cells were grown with certain growth factor, that is TGF-beta and beta-3. And then when this entire biomaterial ink with cells, they are printed and they are cured, then it has been implanted into the animal model. They have shown that there is a cartilage regeneration is possible, or this biomaterial ink can be injected at the defective regions to ensure cartilage formation. As I said before, bioink and biomaterial ink are two different concepts, and these are not synonymous, and you should not use the same phrase or same words. Now, bioink here is, it must contain cells. Biomaterial ink will not contain cells. However, in case of biomaterial ink, you can grow cells on this particular scaffolds when this ink has been formed in the or when the biomaterial ink is printed to make the three-dimensional scaffold. So therefore, we need to be very careful when you use these two terms, bioink and biomaterial ink. And this has been published in one of the perspective review article where my good friend Jürgen Groll from University of Würzburg, Brian Darby, and another collaborator, Michael Galinski, they all put together their efforts to write this particular perspective article. Now, I'll show you two specific examples of our recent unpublished work. First one is towards bone tissue regeneration, and second one is the neural tissue regeneration. In the bone tissue regenerations, we have used gelatin methacrylate based bioinks, but everybody used gelatin methacrylate at the very large concentration, like 10 to 15% gelatin methacrylate. What we are trying to achieve here, initially we started with 10 to 15% gelatin methacrylate, but slowly we have used the lower concentration of gelatin methacrylate, like 5% or 7.5%, and trying to add viscosity modifier and or certain cross-linker, trying to see that whether this biomaterial inks is printable, and then mechanical properties can be significantly increased. At the same time, swelling behavior and biodegradation properties made it more clinically acceptable. Now, the first example for the bone tissue regeneration, we have used the hydrothermally synthesized hydroxyapatite needles. And why needles? Because if the powders are spherical, and then if you're loading 1%, 3%, 5% powders, there is a possibility that is the nozzle clogging. Now, in case of needle separated powders, we expected that it has better flowability, and we use that 15% gelma. And then when you have measured the strength properties, like 15% gelma with 1% hydroxyapatite to 5% hydroxyapatite, you can clearly notice the strength goes from less than 50 megapascal to as high as 300 megapascal, like 6 times increase. Whereas water uptake, which is essentially signature for the swelling properties or mass remaining, that is for enzymatic degradation, here again, it becomes much more sustained. And as you see from the right-hand side, when enzymatic biodegradation was done in the collagenous solution, the gelma hydroxyapatite containing scaffolds actually undergo degradation in a very sustained and slow manner. Now, next stage, as I said, that we have used the gelatin methacrylate with PEGDA, polyethylene glycol diacrylate, and methacrylated carboxymethyl cellulose. This actually was intentionally added as viscosity modifier, and then hydroxyapatite particles second. So this is a typical, polymer chemistry part, that where we have used gelatin methacrylate, methacrylated carboxymethyl cellulose. In the physical gelation, you get uncross-linked. When you do UV cross-linking or use some photoinitiator, essentially the cross-linking takes place, and then gel has a sufficient strength properties. What are the different composition we have seen that you can see in the table, and then bottom plot you can see the strength properties and modulus properties, how they are enhanced with the addition of 1% carboxymethyl cellulose or methacrylated carboxymethyl cellulose. Just to, as far as the numbers are concerned, it can go from somewhere around 5% gelatin. The strength is somewhere around 150 kilopascal to as high as 700 kilopascal. So which is quite high, kind of 4 to 5 times larger strength properties could be achieved. Now, in terms of the elastic modulus properties, less than 50 kilopascal to close to 650 kilopascal, which is almost like 13 times higher elastic stiffness property you can get by addition of the PEGDA, 5% gelma with 5% PEGDA and 1% methacrylated carboxymethyl cellulose. As I said before, that one is to carefully find out the rheological properties as well as the rheology, particularly shear thinning behavior and gel stability. Now, if you look at the right-hand top figure, that viscosity as a function of shear rate, which is clearly telling you about this shear thinning behavior. On the left in these images or left-hand plots, you can see G prime and G double prime, and this is one is the loss modulus, the shear modulus, and there you can see gel stability at 21 degrees Celsius. So there is an increase in the G prime and G double prime values, and field symbols essentially means the G prime, and unfilled symbols essentially means G double prime. So there are certainly some orders of magnitude differences when you go from, for example, 5G to 5G1MC or 5G1MC1H, and 5G1MC1H certainly shows at least 2 to 3 times, 2 to 3 order magnitude higher G prime values and G double prime values. As I mentioned a couple of slides back, that there should be a sharp transition point. And this sharp transition point has been observed also in these our printable inks, where we have added PEGDA and methacrylated carboxymethyl cellulose. And this gel transition window was carefully considered during the 3D extrusion printing of our inks. So every measurements, every physical property measurements give us some clue how to optimize the printing parameters so that we get so that we do not compromise on the safe fidelity. Right. So here again that in terms just 1 second. Yeah, so as I said before, that gel stability is important. And if you look at that, how the G prime and G double prime values are stable, we have found out that almost like up to 15 minutes, that there is not much changes, very significant changes we can observe in the G prime and G double prime values. And up to this 15 minutes, should be the printing time, so that if you prepare the gel and then if you do all the printing experiments over by 15 minutes, we don't expect any problem with these gel properties, and this should be printable. So this is the summary of the sol gel transition, shear thinning, and gel stability properties. Now, with this particular example, I want to emphasize that in the 3D extrusion printing of this kind of gel-based scaffolds, all these properties are to be carefully evaluated. And then 3D extrusion printing experiments can be conducted in a much more scientific, on much better scientific ground, rather than kind of trial and error based level that, changing the printing print bed temperature or the printing speed and so on. So these two properties, as I said before, one is the swelling ratio, and one is this enzymatic degradation. This is the three compositions which we have finally printed, extrusion printed. One is the 5% gelatin, 5G1MC and 5G1MC1H. And there you can see that swelling ratio also significantly reduced by addition of this methacrylated CMC as well as the hydroxyapatite. In terms of the porous architecture of these materials, you can see that as you go from 5% gelatin to all these hybrid biomaterial inks, not only the fraction of porosity is reduced, but also average pore area also slightly reduced. But fraction porosity is largely reduced from close to 83% to somewhere around 67, 68%. And this porosity, fraction porosity, certainly has much better impact on the strength and modulus as well as the biophysical properties which are being correlated in this particular slide. Another things that we carefully consider in doing this swelling kinetics as well as enzymatic degradation kinetics, we try to see that whether it follows certain it follows certain theoretical models. For example, here it is Qt is equal to Q naught plus A into 1 minus exponential minus T by K. And from there, you can find out what is the swelling kinetics. So all this data were carefully fitted into this particular mathematical equations. And from there, we can find out that what is the swelling kinetics. So here you can see that, some of the movies of this, what we have done in the 3D bioplotter in the Envision Tech. The top one is the gelatin methacrylate. Bottom left was the gelatin methacrylate with methacrylated carboxymethyl cellulose. And then bottom right was the gelatin methacrylate, methacrylated CMC, and hydroxyapatite. You can carefully watch it. So you can clearly see the shape fidelity-wise, printability and buildability is much better in 5% gelatin, 1% methacrylated CMC, and 1% hydroxyapatite biomaterial ink. And that kind of a size that we have got, it's 15 cross 15 cross 15, this kind of cube that we are able to obtain. Now let us move to the last one, is a nerve conduit regeneration. Here again, in the case of the nerve conduit regeneration, we have used different printable ink composition, biometal ink composition. So what is the clinical relevance? You can see this is a published study where people have used these autographs for a 41-year-old female patient with adenoid cystic carcinoma of the parotid. And there they have put these donor nerve grafts and trying to bridge the gap there. And this is the typical situation of the autografts, which remains the gold standard, not only in the nerve tissue regeneration, but also for many of the clinical treatment. One of the classic example is that, if for peripheral nerve regeneration, you can cut some part of this leg and you can graft it at somewhere else for the same patients. And this is the concept that I have mentioned to you as the autograft. So potential applications of the gelatin methacrylate ink for nerve conduit regeneration. So this has been attempted by some few research groups where they have put this not only biometal ink or bioink, they printed, and then see that whether neural tissue regeneration is possible in this three-dimensional, in the 3D structures. In our particular case, we have used gelatin methacrylate here, not 5% gelatin methacrylate, but 7.5% gelatin methacrylate, and with PEGDA. And in both the cases, we always use 1:1 ratio. We found out that 1:1 ratio works much better with gelen gum, very small percentage, and carbon nanofibers to make it more electrically conducting. Now, when you develop this biomaterial ink, what we found out that this kind of very hybrid with complex composition of 7.5% gelatin, 7.5% PEGDA, 0.5% gelen gum, and 0.1% carbon nanofibers, slowly this stress strain response actually shifted more from bottom right towards top left. And this kind of transition essentially reflected in terms of magnitude or in terms of values, where you can see strength somehow. So strength is increased from less than 200 megapascal to as high 200 kilopascal to as high as 600 kilopascal, whereas modulus shows much impressive increase from less than 50 kilopascal to as high as 1000 kilopascal, means like 1 megapascal. So almost like 20 times higher. As I said before, that these two properties, one is the swelling studies and one is the bioenzymatic degradation. In both the cases, from pure gelma, like 7.5% gelma, to the hybrid biomaterial inks, swelling is significantly reduced. And at the same time, biodegradation kinetics has been it shows more slow and more sustained degradation in collagenous solution. Now, in terms of the microstructure, certainly the pore area and pore percentage is reduced as we add PEGDA, carbon nanofiber, and gelen gum. It also shows that spores are not that open to the open like in the 7.5% gelatin methacrylate. Now, rheology is important, as I mentioned several times during this talk, particularly for this extrusion printing of these biometric scaffolds. Here again, you see that G prime and G double prime values. You have the temperature sweep, and this temperature sweep distribution, there is kind of rather sharp transition. And in terms of the shear thinning behavior, once again, this biometric ink shows very clear signatures of the shear thinning behavior, which is a most desired property, one of the most desired properties for any printable biomaterial ink. Now, as I said in the beginning of the last part of my lecture, that printability is one thing, but according to clinicians, the buildability is by far more important, because buildability will determine to what extent this biomaterial ink can be printed to a larger structure, which is clinically relevant and clinically acceptable. In this specific case, our long-term collaborator, Dr. Anirudh, a neurosurgeon based in Bangalore, he told us that for most of the neurological peripheral nerve regeneration neurosurgical applications, they need nerve conduit of length around 20 millimeter in length and somewhere around 4 to 6 millimeter in diameter. So we started with the lowest diameter, then went on to increase the outer diameter. So this nerve conduit is outer diameter and inner diameter. Essentially, the difference between these two will determine the thickness of this nerve conduit. But more importantly, length is very important. Now, if you look at this slide very carefully, the lowest one, the 4 millimeter diameter, whether it is gelatin, 7.5% gelma alone, or 7.5% gelma based hybrid ink, in all the cases, 4 millimeter diameter, we could not print to a large structure, maximum up to 5 millimeter in height or 6 millimeter, and also the shape distortion, huge shape distortion is possible. So that also indicates us that 3D extrusion printing has its own limitation in terms of printing large cylindrical structures with smaller diameter. But when you go for the 6 millimeter outer diameter, 8 millimeter diameter, and 10 millimeter diameter, and you can see in all these cases with carbon nanofiber containing biomaterial ink or without carbon nanofiber, with gel gum and 7.5% gelatin and 7.5% PEGDA, in all these cases, we can easily print the 20 millimeter diameter long, 20 millimeter length, large nerve conduit with 6 millimeter diameter, 8 millimeter diameter, and 10 millimeter diameter. And this is how we can see that flexibility of these printed constructs and say fidelity here. If you compress it with a single hand, with a simple fingers, and this can be squeezed, this can be compressed, and then if you release it, it will come back to its original structure. So these materials, we believe that it can be used for the nerve conduits, but certainly we are trying to do a lot more biocompatibility study, including in vivo studies, which is under currently planning. Now here I wanted to mention that this part of the work, we have used a different printer, which was initially supplied by Abhay Biosciences and customized over last 3 to 4 years in our lab with constant input from our students, Soumitra, Jayapriya, and Remya. They have constantly worked with this printer, and they have given a lot of inputs to the equipment manufacturer, that is Abhay Biosciences, and they are able to implement the suggested changes so that this printer can now be used to print this gelatin methacrylate-based hydrogel. And you can see that there is an in situ cross-linking facility is also available. And there is absolutely no shape distortion, and shape fidelity can be ensured. So this is the technology platform in terms of the Abhay Biosciences, and you can see this is their printer, and this is a small movie on this particular printer, which I'm going to play now. Do you want to see this video again, or it is you understood? Everybody? Hello.
Any
Yes, sir,
other-
it is understood, sir. Yes, sir.
Anybody
Okay.
want? Yeah, thank you.
Okay, now I'll show you next 15-20 minutes is that clinical translation, and that one of the clinical study that we have done in 2 different hospitals in India, one based in Bangalore and one based in Wardha near Nagpur. That is what we call cranioplasty surgery. So what we have done, now what is the clinical relevance of the cranioplasty surgery? If a patient has a stroke or road traffic accident injury or intracranial hypertension, it is likely, or the brain tumor has to be removed. Then what happened in these particular cases? There is a depression in the outer side on the outer cranium, and this depression will certainly compromise that aesthetics. And also the cerebrospinal fluid in the brain is also squeezed. So it is not only aesthetics, but also neurological functionality is compromised on those kind of cases. So there are 2 ways you can do things. One is the first is the decompressive craniectomy, like, you remove the tumors. So there is a depression. And after few months, let's say 2 to 3 months, normally neurosurgeons, they perform a surgery called cranioplasty surgery. So it's a surgical procedure which allows the patient to, which allows the surgeon to restore a cranial defect in the or a cranial fault defect after traumatic brain injury, or even after the removal of the cranial tumors. So we have formed a team of neurosurgeons, and then we have done this study in 2 different hospitals, as I mentioned just now. So this is, I'm just going to show you one specific examples, like 62 years old patients who has a road traffic accident. RTS stands for road traffic accident, with left frontotemporal parietal acute subdural hematoma. And this defect size is 12.2 centimeters, a large defect. And then time scale between initial decompressive craniectomy, DC, and cranioplasty is almost like 6 months. Now, when we get the CT scan data, we do a lot of this post-processing using that mesh mixture. And you can see the different views of the CT scan data of the same patient's lateral view, frontal view, and superior view. Now, after careful analysis of these things, it allows you to develop this 3D printing of the skull model. And this 3D printing the skull model, then we do this prosthesis, like we do this polymethyl methacrylate bone flap casting. And after this polymethyl methacrylate casting, this particular flap was freeze dried. And after freeze drying, we do ETO sterilization. And after ETO sterilization, we implant it into the human patients. Now, this is one such surgery where we have used a 3D printed cranium model. After that, we made this polymethyl methacrylate bone flap. And now neurosurgeons are placing this polymethyl methacrylate bone flap in human patients during this clinical study, which we have taken prior approval, not only from hospital IRB, Institutional Research Board, Review Board, but also from Indian Institute of Science Human Ethics Committee. And this bone flap was placed in this, I can show you again, these bone flaps was placed at that particular cranial defect site using titanium screw and using titanium screws, and this can be fixed. This is the right-hand side. You can see what we call in clinical language called intraoperative image. So as you see that, that fitting of the bone flap to the patient is very important. And in this particular slide, you can see that pre-operative, this patient has a large defect, and corresponding CT scan image, you can see that cerebrospinal fluid is certainly squeezed. On the post-operative, after a few months, 2 to 3 months, you can see cranial symmetry is completely restored, and Glasgow outcome score is around 4.2. And post-operative, you can see the cerebrospinal fluid is regained its original shape and volume. What it means, the neurological functionality is retained. And in this particular case, we have found out that maximum defected is 110 centimeters square. So this has attracted a lot of media attention, and then we normally meet these clinicians, and then you can see people from Abhay Biosciences also present in this national conclave, which we organized more than a year ago. So what I did not show you in this presentation, our work on the dental implants and hydroxyapatite powder synthesis and hydroxide-based materials, and also urological neobladder reconstructions, where we have used thermoplastic urethane polydimethyl siloxane blend for neobladder reconstructions, and also our work in the bioengineering, where we do significant study on the electrical field stimulation, magnetic stimulation to guide the stem cell functionality. Now, what we believe that, that this India needs indigenous manufacturing capability of these biomaterials and implants. I'm very happy to let you know that, that I could inspire the Tata Steel New Materials business, and they have established a new business vertical called Medical Materials and Devices 2017, and they started marketing, selling already thermally sprayable hydroxyapatite. Now, in order to develop a vibrant translational ecosystem, I have 6-fold recommendations, which I have written. Based on these recommendations, I have written a book which was published by International Science Academy and Springer Nature. And these 6-fold recommendations are as follows. One is related to the national regulatory framework. Second one, basic research strengthening. Third one is a clinical context defense translational research. Fourth aspect is the industry collaborations and capabilities. Fifth one is the medical education and clinical research revamping. Sixth one is the national policies implementation and implementation. So in terms of the clinical context-driven research, what I believe that India should have more GLP compliant biocompatibility testing and GMP compliant manufacturing facility. Also, it should have preclinical more testing institute like IVRI, Indian Veterinary Research Institute like institutes, where many of the biomaterials and implants can be tested. In terms of the industry collaborations, we should have more pilot scale production facilities for different biomedical grade hydroxyapatite, titanium alloys, cobalt chrome alloys, UHN22P. These are like really important. And we should also have Industry 4.0 in place in the biomedical manufacturing sector. So one of my key recommendations is called what we call biomaterialomics. In the biomaterialomics, essentially what we believe that it can serve as a good platform to allow the convergence of the biological omics as well as the additive manufacturing related attributes. In the biological omics essentially means metabolomics, genomics, transcriptomics, proteomics, or the additive manufacturing related attributes like binder formulation, biocompatibility testing, printing parameters, and so on and so forth. That what we have done before. These are the key elements of this biomaterialomics. One is the biomaterials development, like different biomaterials library, biocompatibility assessment, different first principle calculations, either density functional theory or in silico modeling, like molecular dynamics simulations or finite element analysis, preclinical studies. There are several databases or e-platforms are available, or online platforms for clinical databases or preclinical data, or CellNet, like for stem cell based data are available in certain online resources. And finally, we have to utilize artificial intelligence and machine learning based algorithms, like machine learning, deep learning, depending on what is the quantum of data available, to accelerate this particular biomaterials development and innovation. In the specific case of organ-specific tissue engineering, in the pre-printing level, there are different parameters which are important. In the 3D printing space, like optimization of printing parameters and printability and buildability, how these different rheological properties are important. Post-printing, like optimization cell culture protocol, quantitative analysis of safe fidelity, these are important. So this work is recently published in a review article published in Acta Biomaterialia. So over the years, I have been able to develop these interdisciplinary and translational ecosystems by bringing innovation ecosystem closer to that of the clinical ecosystem and manufacturing ecosystems, where I have been constantly working with the government agencies and private sponsors, as well as different professional societies like Biomedical Society in India, or International Union of Societies for Biomedical Science and Engineering, International Academy of Engineering, American Ceramic Society, and so on. So in next generation researchers in academia, many startups, some of the established companies, their personnel are being trained. And this has been a large source. This has really created a more vibrant translational ecosystem here. So for many teachers and students, I would like to refer some of our books. And I would like to acknowledge generations of the PhD students, project assistants, research associates, former students who are currently serving as faculty members in some of the IITs or NITs, or as postdocs in different reputed laboratories around the world. They really contributed to the growth of this particular area in my group. And thank you very much for your kind attention.
Thank you, sir. And now the floor is open for the questions. Audience, you can ask if you have any doubts. Audience?
Okay.
Yeah.
Ravi?
So, yeah, I don't know. So
Good evening, sir. I'm Amay here. I have one doubt. Sir, so the print is there a printability window which I have seen for the piston-based printer and the pneumatic printer? So so how will it change? Like which one will have more printability in terms of which type of 3D printer will be better? Extrusion 3D printer, like
you mean to say accuracy level, which is better?
No, like printability, like higher range of
It is very difficult to answer these kind of questions because it all depends on your biomaterial ink, right? So when you want to use one specific biomaterial ink, then that biomaterial ink you have to use in that, whether pneumatic or pressure assisted or screw based 3D printer. Then only you can say which machine has better printability. So if you change the biomaterial ink, automatically the printability will be different, right? And unfortunately, these kind of control studies are not available in the published literature.
Thank you, sir.
Okay.
Yeah, thank you, sir.
Any other people? Any other doubts? Naidu or Kishore? You have any doubts?
Hello, sir.
Sir,
Myself.
no, sir.
Krishna, sir.
It's okay for me.
Yeah. Please go ahead, one by one. Yeah, Venkat, you can go ahead, Krishna.
Hello, sir. Sir, how we are evaluating the elastic properties of Naru conduits, sir?
Elastic properties means that you have, we do the compression.
Tensile properties.
No, we do compression testing for all these things. We can do micro UTM just to do compression testing. So you cannot do standard UTM because standard UTM is meant for metallic samples or ceramic samples. For polymeric samples, the gauge length and then all these sample dimensions are also much less.
Okay, sir.
Sir, good evening, sir.
Yes.
Sir, this is Kishore from Goodlowly Engineering College, sir.
Yeah.
Sir, for scan 4, if the pore size it increases the porosity structure, sir.
Yeah.
Pore size it influences the porosity structure.
Yeah. What's your question?
Sir, my question is the pore size influences the porosity structure?
I did not get your point. If pore size increases, the porous structure will increase, right? Pore area will increase. So what is your
question? It's a halfway question. Can you repeat again? If
the pore size influences the porosity structure.
Pore size influences the porosity structure.
Of course,
Yes, sir.
of course, it will influence.
Of course, it will influence. Now, if you want which type of influence or something, that is all depend on what type of post thing.
Regarding this strength, sir.
Strength will certainly, yeah, strength will be compromised. Strength will be reduced, right? If pore size increases, yeah.
Your density is going to reduce. So obviously, your strength will go to reduce.
Sir.
Yeah. Please go ahead.
Yes, sir. Is the strength also influences, sir?
No, it will decrease. No? That is what professor saying.
Yeah. It decreases, sir.
That is what the common tendency.
Okay. Sir, seen in one paper, sir. For example, if you take a rectangular cube structure, okay, and if you do the analysis in using any additive manufacturing, it will design the cube into a not with full of material and use the same strength to the between these tools.
So that is different, I think. So what you are talking about is lattice structures in metals. Okay.
Yes, sir.
So lattice structures in metals is different, and scaffolding structure of pores, what professor is taught is slightly different. I think professor can answer that.
Yeah, you are right. Yeah, basically, I think he's confused between this lattice structure and then this scaffold structure. Yeah.
So like I section and other things you'll know. You see in strength of materials, when I structure is normal complete solid will be replaced by I section will have similar like strength analysis and other things. Similarly, lattice structures also plays a crucial role nowadays. So those are different, and professor, what are the pores and other things he start in scaffolds are different actually.
Yeah. Okay, sir.
Okay, thank you.
Thank you, sir.
So with this, so 9 to 9:30, we have the examination today. So with this, I think Professor, thank you, sir, for your valuable time. And in spite of his busy schedules, I think from Germany to UK, UK to, I expect Professor is either in Germany or in UK. I don't know. I still don't know, because when I call him, he says, I'm going for Germany to UK. So I think he's in Manchester, maybe. Professor, you are in Manchester now?
No, in Germany.
Okay, so you see that I said no already. So yeah. So, thank you, Professor. And,
Okay, thank you. Bye.
yeah. So I thought I want to invite, when I called, I want to invite you in person for next week, physical one. So then you said that your phone is not going, Indian call is not going. Then I put into WhatsApp. So you said I am in abroad. So then I don't have. Okay. So next time, Professor, I think you have some time for us.
Sure. Okay. Thank you.
Thank you, sir. Thank you, sir. So now, Venkat, are you there? Venkat?
Yes, sir. Yes, sir.
Yeah. So we'll start the exam.
Yeah, yes, sir.
Where Uday is there?
No, sir. Uday is not.
Who is going to start the exam now? Yeah, please, you can have a 5 minutes break before we start the exam. It is just a basics. Don't worry. So you can inform in the WhatsApp group to all the people saying that examination we are going to conduct. So let's break for 5 minutes. We'll start exam at 9.
So shall we leave this platform, sir?
Venkat, I think exam is here only. I don't know.
No, sir. We will share the
Google
link for
Form.
them. Google
Yes.
Forms.
Yes, yes. So we will leave, sir.